Thank you, Joel, for the kind introduction and for the invitation. It's great to be here. My thanks are for the Cohen Foundation to organizing this and supporting a big effort in Lyme disease, which has been sorely needed. So what I'll tell you about today is about our work in a variety of infectious pathogens that is related to the general problem of treating infectious diseases, and of course about our data with Lyme disease. Okay, so for starters, I want to tell you that most chronic infections are caused by pathogens that are susceptible to antibiotics. And that is the main paradox of chronic infections. If it doesn't make any sense to you, that means that you are wide awake, because paradoxes make no sense. That's the definition. And what we found a number of years ago is that in, let's say, a typical chronic biofilm infection, the majority of cells are highly susceptible to killing by antibiotics, and then you hit upon this small subpopulation of persister cells, which are essentially invulnerable. So that, that resolves the paradox. They are not resistant, they're not growing, and they're not dying in the presence of pathogens. So it turned out that these are dormant, spore-like cells. That explains their tolerance to antibiotics, which kill by corrupting energy-dependent functions. And so what here, I'll give you a summary slide, a little over a decade of work from our lab. And what we found, first of all, is that the pathways of persister formation are highly redundant. And if there is a general theme in this case from the model organism E. coli, it is toxin antitoxin modules of different sort that are persister genes and are responsible for persister formation. So these, for example, inhibit protein synthesis that causes dormancy. The interferase type toxins cleave mRNA, that causes dormancy. And then this pathway, which was described by Toby Dorr, a grad student who's now at Cornell, although not in New York, right, on the undergraduate campus. He's a faculty there. So he found that the DNA damage SOS response actually turns on, apart from DNA repair, turns on this very interesting protein TISB, which is an ion channel, decreases partially the proton motor force in ATP, and that causes whole systems shut down and dormancy. And that will be especially relevant to what I want to say today, as you will see in a moment. Okay, so armed with that information, we decided to see what happens with persister formation in gram-positive bacteria. We took Staph aureus. It also has toxins, antitoxins, only 3 of them. We knocked them out. This is work by Brian Conlon, who's now at faculty at Chapel Hill. And what he found is that here's a stationary state killing with antibiotic. There's absolutely no difference between wild type and toxins, antitoxin knockout. It's the same in exponentially growing cells. So what we learned over a decade of work was completely irrelevant, right? So for understanding persisters in gram-positive bacteria, so we had to start from scratch. And this is what we decided to do. We had, for a variety of reasons, a suspicion that persisters may be cells that are going into stationary state early. And here's a test of that hypothesis. We have this promoter of arginine deaminase, which is expressed in stationary state. You see it's highly expressed in stationary state, poorly expressed in exponential, but there is this small subpopulation of cells in exponential that has stationary levels of expression of that promoter. So we sort out these cells, and these are the only ones that survive a hit by antibiotic. So they are the persisters. And then, of course, the interesting question is, what is it that this promoter is actually measuring? What is it that it is responding to? Okay, so we decided that it responds to ATP. I don't have time to tell you why we decided that, but this is what it does. If you decrease the concentration of ATP using arsenate, then this promoter and a similar one, PKA, shoot up. So actually, we are measuring ATP or the low levels of ATP when we're sorting out persisters. So stochastic variation of ATP apparently is what's responsible for persister formation on staph, and this is very relevant to Borrelia burgdorferi because it has no toxin. It is one of the very rare bacteria that have no toxin antitoxin modules to begin with, so they have to make persisters by a different way, not dependent on toxin antitoxins. So then we decided to revisit what we know about E. coli. We found a similar marker in E. coli that responds to ATP, in this case positively responds. It's the ribosomal promoter. We sort out bright cells, we sort out dim cells in this case, and the dim cells are the only ones that survive a hit by antibiotic. So here we have a revision of what's happening in persisters. It looks like, apart from these specific pathways, which are probably very different in different bacteria, there is this general pathway, which we're now, of course, investigating in Borrelia as well to see if it operates in Borrelia, as it is in unrelated bacteria. And we are, of course, on the hunt for factor X that is responsible for dropping this ATP. Next time, I'll probably tell you what it is. Okay. So another important question regarding persisters that we've been addressing is clinical relevance of persisters to disease. It is hard to do Koch postulates on persisters because a small subpopulation of temporarily dormant cells, you put them in an animal, they wake up, they're not persisters anymore. You cannot do Koch postulates on persisters. So a clue to finding a link between persisters and disease came from an experiment that Pooja Balani, a grad student in my lab, was doing on a very different subject. She was trying to catalog all potential persister genes. And here's a simple experiment that allows you to do that. So you hit a growing population with antibiotic, you collect surviving cells, repeat it a couple of times, and you get an enrichment in mutants, high persister mutants, that make more persister cells, right? Then you can sequence the genomes and find out what those mutations are and what the genes are. So, but then when you look at this experiment, what comes to mind is that this is exactly what happens when patients are treated with antibiotics. They receive periodic high doses of antibiotics, and if being a persister is useful for the pathogen to avoid being killed by antibiotic, you will get selection for high persister mutants. So then we went hunting for such mutants in patients with a relapsing UTI, because we figured that this experiment has already been performed in humans on a massive scale. We just need to look at the results of that experiment. So I'll show you what we found. What we found is primarily in E. coli, a gain of function mutation in the hip A protein that makes it more active and it more efficiently shuts down translation and causes dormancy. And here are these mutants. They make 100 to 1,000 times more persisters than the wild types. So that links persisters to disease and also shows you that there's a separate phenomenon out there. Apart from heritable resistance, there's heritable drug tolerance. Two completely different phenomena, both employed by pathogens to avoid being killed by antibiotics. Okay, and then we decided to check this out in Borrelia burgdorferi. Now, so I asked Lyndon Hugh, my collaborator, to give me a collection of Borrelia isolates from patients that have been treated with antibiotics. He said that's not possible for a variety of reasons, many of which you here appreciate. So then we decided to do something different. So from our investigation of E. coli, we were surprised to see that persister mutants come not only from patients treated with antibiotics, but also from commensals. So it looks like in Mother Nature, there are lots of nasty compounds that hit the pathogen and select for high persister mutants. You don't need antibiotic treatment in the clinic to select for them. So then we were thinking maybe the same thing may be happening with Borrelia that infect ticks. And so Sam Telford gave us a collection of Borrelia that we're screening now for the presence of high persister strains. And this is what we're finding so far. So this is the degree of killing by ceftriaxone of these isolates. And you see that this one, for example, is killed very well, but not these. Okay, so this is the same kind of a pattern that we see in E. coli, and we see in all other pathogens that we investigated and published so far. So we published this in M. tuberculosis, Pseudomonas aeruginosa, Candida albicans yeast. So it looks like this is a general phenomenon. This may be very relevant to the disease because one of the things that we will be discussing this afternoon is what is PTLDS and how you can predict and link initial infection to who will and who will not develop PTLDS. Well, this is one possible variable that you have. Some people are infected with an easy form of the pathogen and some with a form that will be more difficult to eradicate. So now I want to, from here, want to tell you about the compounds we've been working on in a variety of species to eradicate persisters. I'll start with one compound that we identified, and this is in an effort to eradicate biofilm infections of Staph aureus. Now, eradicating persisters, as you may imagine, is a difficult proposition because these are dormant cells. As I told you, they're not killed by existing antibiotics. And the pathways of their formation are redundant. That means you don't have a drug target, right? So it looks like a perfect dead end for us. But one thing that comes to mind when you see that nature came up with a perfect defense, nature usually comes up with a way against that perfect defense, right? So we started looking among natural products, asking the question, did Mother Nature bother to evolve an anti-persister compound? Knowing what we're looking for a compound that's going to hit an important target in the cell, corrupt it, and kill that cell, and that will require no energy, right? That's the magical compound that we're looking for. But by these criteria, we hit upon this old abandoned antibiotic, acyl depsipeptide. Or ADEP. It does an interesting thing. Its target is the CLIP protease. The CLIP protease normally recognizes misfolded peptides with the aid of this ATP-dependent chaperone, and then it digests them. In the presence of ADEP, the pore of the protease is open, and it can digest peptides without the need for ATP, and that's exactly, of course, what we're looking for. So Brian Conlon investigated this, did a big study, showed by proteome analysis that there's a massive degradation of mature proteins in the presence of ADEP. In Staph, I'll just show you one or two key experiments. Here's a control with stationary Staph aureus, growing Staph aureus. Here, persisters that are not killed by antibiotic. And if you add ADEP, you get complete sterilization. So that was the first time we saw sterilization of an infection in a test tube, and that was pretty exciting. And then, of course, we did that in an animal model of incurable biofilm infection. This is a neutropenic mouse. Incurable thigh infection, regular antibiotics don't do much. ADEP completely sterilizes. We add rifampicin or another partner antibiotic to prevent the rise of resistant mutants. And so we got a sterile mouse, and this we are developing now. Hopefully it will move into the clinic. So this is encouraging, but unfortunately ADEP has no activity against Borrelia. So here we had to start again from scratch, as you often start in microbiology. And we were looking for vulnerabilities of this pathogen. We have ourselves a list of vulnerabilities. And one thing that is helpful is that the genome is small, so it doesn't have a lot of tricks up its sleeve. The Borrelia actually doesn't with its small genome. So one thing that we were surprised to find is this. If you look at persister formation so here's the growth of the culture. So it grows exponentially, then it reaches stationary, non-growing state. And during the time of growth, you can take samples exposed to antibiotics and find out how many cells survive. Those are your persisters. Persisters shoot up from early exponential through mid-exponential. So here the culture stopped growing, right? And there's considerable killing, especially at the earlier stationary state, by amoxicillin and by ceftriaxone, enormous killing of stationary state. These are cell wall-acting antibiotics. They're not supposed to kill non-growing cells. We've never seen that before, with any pathogen. So we were very surprised. Why is it that cell wall-acting antibiotics are killing an ungrowing cell? So we decided to investigate this. This is work by Wendy Wu in my lab. She's probably somewhere here. So here, first of all, you see how different antibiotics kill stationary cells. Doxycycline, ceftriaxone, much better. Vancomycin actually is a terrific anti-Borrelia compound. Okay, so that is also something to keep in mind. But it does not sterilize. But still, enormous killing. So here is an experiment showing you label incorporation, fluorescent label incorporation into peptidoglycan. This is growing Borrelia. The green label incorporates, of course, especially you see lots of label at the ends. And this is stationary. And you see that there is significant label incorporation into stationary cells. Here's the middle of the cell. So in the middle of the cell, lots of incorporation of peptidoglycan. This is an ungrowing culture, but it continues to synthesize peptidoglycan, and that's apparently why it is vulnerable. Here is a quantitative measure of that same experiment. So here is, by comparison, Staph aureus. Growing Staph aureus, very little label incorporation to stationary Staph aureus. And here's Borrelia. Here's Borrelia log. Not that much of a difference in Borrelia stationary. So here is the interesting vulnerability, the anomaly of this pathogen that we should definitely exploit. Okay, so the other thing that we've been thinking about is how can we eradicate persisters completely with something like a cell wall-acting antibiotic. This is work by Bijay Sharma. She's now a postdoc in Lyndon Hughes' lab. So the simple idea was to hit the population with antibiotic. Persisters survive. And then you wash away the antibiotic, allow persisters to wake up, but not to repopulate the population. And you hit it with antibiotic again. And of course, since they woke up, there are less of them. So you get a drop. You repeat it 2 more times, and you get complete sterilization, right? With a compound that itself does not sterilize. We did that because, of course, we were thinking that in the body, the drug will be cleared from the system, and if you can hit upon the correct pulse dosing, you can probably eradicate a chronic infection with a compound that does not necessarily itself sterilizes. So we're now doing this experiment in mice, and we're following to see if we achieved complete eradication, and hopefully, if that works, we can do that in humans as well. Okay, apart from that, the last thing that I'm going to tell you about is that we have also a drug discovery project. This is also a work that is performed mainly by Wendy Wu. So we have a screen for compounds that inhibit growth of Borrelia. We started with a pilot screen. Now we screened a couple of several such pilot screens now make up a bigger one. And our initial hits were, reasonably 0.45% hit rate. So we were looking for compounds that specifically act against Borrelia, don't act against other bacteria. If you do that, that gives you an immediate advantage, because then that is a strong indication that your compound is not going to be toxic, because we usually do not have that kind of a target. So with that logic, we found something interesting. Apart from these 20 compounds, we found this compound. It also showed up and was published recently in a screen that was performed by the Stanford group of Rajadan and his co-authors. So here's this compound. This is disulfiram. Normally, disulfiram is used to treat alcoholism. Well, it doesn't treat it, right? But it kind of prevents people from drinking alcohol. Right, because it's anti-abuse. It's called anti-abuse because it inhibits this step in ethanol degradation. On the way from ethanol to acetate to carbon dioxide and water, there is this nasty compound acetaldehyde, which is toxic, and it is detoxified by acetyl dehydrogenase, acetaldehyde dehydrogenase. And that's what the disulfiram inhibits, so you get a horrible hangover. And, you know, it's unlikely that you're going to drink alcohol a second time. Disulfiram is highly active against Borrelia burgdorferi, has no activity against other pathogens that we tested, so it's specific against Borrelia. And here, if you look at this ranking of antibiotics, here is very interesting. Disulfiram completely sterilizes Borrelia burgdorferi. It kills persisters. We are looking into the mechanism of this. This is quite unexpected. But the compound is, of course, approved. It's very safe. And so we're gearing up to do mouse studies to see if we can cure a mouse of Lyme disease. And then this becomes a very interesting candidate to treat Lyme in people. Okay, so with that, thanks to my team, to my collaborators, and to support, and I thank you for listening.
I'll be very loud. Jim, could you perhaps hypothesize as to why Borrelia is particularly targeted with this pathway, with this drug? Does it, you know, what is it? Is it alcohol producer or something?
That's an excellent. That's an excellent question. Well, it is an anaerobe, so maybe that's why it's particularly vulnerable. Interestingly, it does not have a recognizable homologue of acetaldehyde dehydrogenase. Of course, that's the first thing that we checked. So our simple hypothesis is that it probably has an enzyme that we did not recognize in our database. So, but we'll figure out what it is.
So, I'm sorry. So the question was about low-dose penicillin administered as weekly bicillin injections. Sometimes we see failures of IV antibiotics and other regimens, and all of a sudden we switch the patient to a single injection a week, and the patient recovers. And the question is whether in all this hunt for the persister killing, whether there is any suggestion that there is a chemodetection, some sort of chemodetection by the microorganism which enables it to transform into a persister, into a, we know that there are naked forms that are dropping the cell wall, therefore the cell wall antibiotics won't work.
Yeah.
And the question whether this low dosing evades the chemodetection and therefore might offer a effective killing and prevention of forming persisters.
Yeah, it's a good question. So one thing that you notice from these biphasic killing curves is that in the presence of antibiotics, persisters do not die. Once you take away the antibiotic, add fresh media, allow them to re-add antibiotic, they do die. The fact that they do not die in the presence of antibiotic means that they're not waking up, right? So they are probably sensing the presence of antibiotic, and paradoxically, the antibiotic prevents them from effectively waking up. And that's definitely one of the things that is potentially one can exploit to find out what that is and eradicate persisters.
Just to go on that question of the disulfiram, do you see any difference between the B31 and any of the clinical isolates, either with disulfiram or some of your other drugs as well?
I'm sorry, between what?
B31 and clinical isolates?
For disulfiram. We haven't done that yet. So we definitely are planning to test our whole panel with disulfiram.
I have a question on as we've seen with other microbes, the influence of, in particular, thinking about the case of E. coli, where a bacteriophage in one case was dropping in things like methyltransferases and reprogramming E. coli to become virulent and things like that. Do you see, or have you seen any evidence of the other part of the microbiome, the viruses or bacteriophage, adding some variability to either whether resistance or virulence or anything like that?
In Borrelia.
In Borrelia.
Yeah, we have not seen it. Again, it keeps a very small genome. It does not take up foreign DNA very easily. For example, I don't think that anyone has described Borrelia that carries a transmissible drug resistance marker. Determinant. And then another thing about Borrelia and resistance that is useful to keep in mind is that the disease is apparently not transmissible between humans. So that means that every human is a dead end in a way. So even if with a low probability resistance will develop in that patient, it's not going to be transmissible, because it is the transmission of resistance that is the main problem for current drugs. And that's why I think we can, with relative impunity, think about, pulse dosing antibiotics and not worrying too much about the probability of resistance spreading.
Have you tested any of the differences in geographic strains?
Not yet, no. But if you have a collection, I will test it.
That's actually a good lead into my next question was, so we have clinicians and people collecting big samples, and from your perspective, based on what you showed, or maybe something you didn't show, if you had an iPhone app that made Mark do whatever you told him to do, or anyone else, what are the experiments that we should be running, maybe as a consortia or something, that aren't being run right now, but from your perspective need to be done? Can you do anything come to mind?
Well, the simple survey of the relative level of persisters in different isolates, that would be very nice to know. How prevalent are apparent high persister mutants, and then can we link that causally to the probability of developing PTLDS? That would be a terrific question to ask. Another good question to ask is any experimental or very highly effective drug that we see that eradicates Borrelia, like vancomycin or disulfiram, how broadly applicable that is to a large number of strains.
Yeah.
Have there been any, based on the last results that you were indicating, have there been any attempts to use disulfiram, which I believe is available, it can be, to use it in clinical populations and see what happens?
Well, that's exactly what we're gearing up to. So we're running a mouse study. First of all, we want to see if it cures uncomplicated acute infection in the mouse. Yes?
I guess I meant not that you were doing directly through mouse studies and the normal progression there, but individuals who are having serious problems with Lyme, and it's a relatively safe drug, I believe, although I'm not.
Yes, it is fairly safe, yes.
So are individuals separate from you attempting to see whether it has an impact clinically before the animal studies are completed?
That's an interesting question. Sure, one could conceivably, put it right into people. Why bother with mice? Yeah, you could do that. We're also running a retrospective study with Chris Patel. with Chirag Patel from Harvard School of Public Health. So he has access to medical records of 20% of American people. He's actually not unique in that. Just keep in mind that that information is out there. And so he is asking a question about correlates between Lyme disease and a number of other things, including, of course, whether taking disulfiram is good for you when you are bitten by a tick.
I was wondering a little bit about persisters. So how specific to a given environment is a persistent strain? I mean, so for instance, if you're a persister in, I don't know, in one host, under a certain antibiotic condition, would you be persistent in another host as well, for instance?
Right. Well, the one important factor that determines the level of persisters is the growth phase. There's almost no persisters formed in early exponential, and then that probability increases about 1,000-fold as the population approaches stationary. And that's a universal determinant, and it looks like all other determinants are relatively minor by comparison to that one.
Okay, so if I understand you properly, then basically, if you're a persister in one environment, for instance, a persister in, in an animal model, you'll also be a good persister in a human or human cells,
Yes.
or that sort of thing. Okay.
Dr. Lewis, often patients are infected with more than one organism, and that seems to also be a cause for ongoing symptoms and difficulty with the cure. So I'm wondering, did you consider testing mice with not just Lyme disease, but adding another pathogen to see if the disulfiram worked? Thank you.
That's an excellent question. We're starting with something simple. So Joel likes to work with massive data and very complex systems. I like to work with one thing at a time, right? So probably that's why we're collaborating with Joel. But we'll certainly add that once we figure out what's happening in a simpler system.
Thank you.
Not every day we get the opportunity to have somebody of your caliber talking to clinicians, and there are, several perhaps mythologies in Lyme treatment, and I want perhaps your authoritative statement to break down those mythologies. For example, the Lyme community has been talking for ages about cysts and cyst busters. Are these the persisters?
Yeah, that's a complicated question. We are investigating that now, but I think we have also an afternoon panel where we will be discussing, things like you are alluding to, breaking down barriers, and what are really the questions, the burning questions, or the unanswered questions that we need to attack. So we'll probably do all that later today.
Thank you.
Since we have a little time, I guess I got another question on here. From the work, I know you focus very much on the microbe, but what sort of hints or hypotheses are coming up in terms of even from a drug administration standpoint, modulating the host immune system, or the role of the host immune system in the progression of the disease?
Yeah, sure. So we gave that a considerable amount of thought. Modulating the immune system directly is very tricky, but there is a very good and fairly benign way to modulate the immune system, and that is through altering the microbiome. And again, one of the things that I would like to discuss this afternoon is microbiome intervention into autoimmune disease type presentation.
As a corollary to that question, what is the impact of the immune system on the development of persisters? Specific side?
Well, I would say that so one thing that the immune system does, the innate immune system produces reactive oxygen species. So we've shown in vitro that, at least in E. coli, reactive oxygen species can induce persister formation. There's been a belief in the tuberculosis field that the immune system keeps persisters in check because, if you have latent TB and then you get, let's say, HIV, you get a flare-up of the disease. So that is indirect information on at least that case of latent disease. But apart from that, not that much is known.
Thanks.